morning and welcome to Biology. I'm Paul Suchecki and this is my classroom. It's great to see all your smiling faces again today. Today's topic is genetics. Have you ever wondered why you look like your parents or sort of like your brothers and sisters? My dad says I look like the mailman. But anyway, genetics is the study of heredity, and heredity is the passing on of traits from parents to offspring. Humans have had a basic understanding of genetics and heredity for thousands of years. Even back then, people understood that something was passed on from parents to offspring generation after generation. They never looked in a microscope. They never saw a cell. They never heard of chromosomes or DNA. They saw the same thing in their animals and in their crops. Something was passed on. In the beginning of agriculture, farmers knew that something was passed on from generation to generation. They understood seeds, and they understood the value of hardy plants. They weren't only early farmers, they were early geneticists. In the 1860s, an Austrian monk named Gregor Mendel was the first person to conduct formal scientific experiments to study genetics. Mendel grew pea plants in the monastery garden to discover how traits like seed color and seed texture and plant height were passed on. Mendel did brilliant science and kept detailed records, but unfortunately, his work went mostly unnoticed. Modern scientists appreciate the contribution that Mendel made to the field of genetics. He laid the foundation for all the current and future discoveries. Today, Mendel is known as the father of genetics. Who's the mother of genetics? Mendel discovered that traits like height of a pea plant are controlled by what he called factors. Today we call these factors genes or alleles. Remember, Mendel never heard of DNA or chromosomes. Some genes are dominant and some genes are recessive. When a dominant gene is paired with a recessive gene, the dominant gene is expressed. The recessive gene just kind of goes along for the ride. It's basically silent. Usually the recessive trait is only expressed when two recessive genes are paired together. Mendel cross-pollinated a true breeding tall pea plant with a short pea plant and was surprised to get all tall offspring. Then, when he cross-pollinated some of the tall offspring from the first cross, about 75% of the resulting second generation were tall, but 25% were short. So basically, he crossed tall with short and got all tall. Then he crossed tall with tall, and he got mostly tall, but some short. Huh? That's a head-scratcher. Turns out that in pea plants, the gene for tall stems is dominant over short stems. Let's use a big T to represent the tall dominant gene in pea plants. We'll use a little t to represent the short recessive gene. Genes always come in pairs, remember? Like shoes. The genotype is the genetic makeup of an organism. The genes, the letters. The phenotype is the outward expression of the genotype, what it looks like. In this case, is it tall or is it short? In Mendel's first cross, the genetic makeup or genotype of the tall plant was big T, and the genotype of the short plant was little t. We'll call this the P generation or parent generation. Big T crossed with little t. We'll use a Punnett square to show the outcome of this cross. We start by separating the two genes of the first parent and placing them here and here. We'll separate the two genes for the other parent and place them here and here. Now we fill in the boxes where the rows and columns intersect. In this box, we bring this one down, this one across. Big T, little t. We'll bring this one all the way down and this one across. Big T, little t. We'll bring this one all the way across and this one down. Big T, little t. We'll bring this one all the way across and this one all the way down. Big T, little t. So it turns out that the genotypic ratio for the F1 or first generation offspring of this cross is 4 big T, little t. And nothing else. The phenotypic ratio, all four are tall. Well, that's what Mendel got. He then crossed two of the F1 offspring plants to get a second generation, or F2. So now our cross looks like this. Big T, little t, crossed with big T, little t. Like before, we draw our Punnett square, 4 boxes. We'll separate the two genes for the first parent, they go here and here. We separate the two genes for the second parent, they go here and here. Now we fill in the boxes. We bring this one down, this one across. Big T. We bring this one all the way down and this one across. Big T, little t. We bring this one all the way across and this one down. Big T, little t. And we bring this one all the way across and this one all the way down. little t, little t. The resulting genotypic ratio is 1 big T, 2 big T little t's, and 1 little t, little t. Our phenotypic ratio, 3 tall, 1 short. Notice that 2 different genotypes produce a tall plant, big T, and big T, little t. That's because the big T gene for tall is dominant, and the little t gene for short is recessive. When the 2 genes in an organism that control a trait are the same, we call that homozygous. When the 2 genes in an organism that control a trait are different, we call that heterozygous. Homozygous, same genes. Heterozygous, different genes. But there's no noticeable difference between a homozygous tall, big T, and a heterozygous tall, big T, little t pea plant. So if you had a homozygous tall pea plant and a heterozygous tall pea plant growing side by side, you wouldn't be able to tell the difference just by looking at them. Even if you took out a microscope and looked at a cell from each plant, you still wouldn't be able to tell which one was homozygous and which one was heterozygous. They're both tall. There is one way that you could tell. Run what's known as a test cross. A test cross is when you cross each plant with a short recessive plant. If all of the offspring come out tall, then the plant was big T tall. If about half the offspring come out tall and half the offspring come out short, then it was big T, little t tall. When it comes to monohybrid genetic crosses, those involving just one trait, there are really only 6 possible crosses. Big T crossed with big T, big T crossed with big T, little t, big T crossed with little t, little t, big T, little t crossed with big T, little t, big T, little t crossed with little t, little t, and little t crossed with little t, little t. For number 1, I think the genotypic and phenotypic outcome is pretty easy to predict. Big T crossed with big T, all the offspring are going to come out big T, and they're all going to be tall. How about number 6? I think that one's pretty easy to predict too. Little t crossed with little t. Do we even need to do a Punnett square to predict the outcome? I think all of the offspring are going to come out little t, and they're all going to be short. So that really only leaves us with 4 crosses to solve. We already did number 3, big T crossed with little t, and we already did number 4, big T crossed with big T, little t. So let's have a look at number 2. Big T crossed with big T, little t. We set up our Punnett square. We separate the genes for this parent. We separate the genes for the other parent. Now go ahead, fill in the boxes. Big T, big T, big T, big T, big T, little t, big T, little t. For our genotypic ratio, we get 2 big T and 2 big T, little t. For our phenotypic ratio, all 4 are tall. Check. That leaves us with number 5. We're going to cross heterozygous tall with short. Again, let's use a Punnett square to predict the outcome. For our genotypic ratio, 2 big T, little t's and 2 little t's. For our phenotypic ratio, 2 tall and 2 short. Probability is the likelihood that an event will occur. A Punnett square is a tool to predict probability. In a Punnett square, there are 4 boxes, so there are 4 possible outcomes. In cross number 4, big T crossed with big T, little t, one of the boxes has big T. So there's a 1 out of 4 or 25% chance of getting big T. Since 2 of the 4 boxes are filled with big T, little t, that's a 2 out of 4 or 50% chance of getting big T, little t. And since 1 box has little t, that's another 1 out of 4 or 25% chance of getting little t. So if this genetic cross, big T crossed with big T produced exactly 100 offspring, it would be a safe bet that about 25% of them would be big T, about 50% of them would be big T, and about 25% of them would be little t. And it would be a safe bet that about 75% of them would come out tall and 25% of them would come out short. Not all genes are dominant or recessive. In some cases, differing phenotypes could display incomplete dominance or codominance. For example, in Japanese 4 o'clocks, they're plants. They come in 2 colors, red and white. Red is not dominant over white, and white is not dominant over red. They show incomplete dominance. The 2 traits blend. When you cross a red with a white, you get pink. We'll use a big R for red. We'll use a big W for white. Red's not dominant nor recessive. White's not dominant nor recessive. They're incompletely dominant. RW is pink. Since this also is a monohybrid cross, there are really only 6 possible combinations. RR crossed with RR. RR crossed with RW. RR crossed with WW. RW crossed with RW. RW crossed with WW. And finally, WW crossed with WW. Try those. See what you come up with. 1 and 6 are pretty easy to predict. For number 1, cross red with red, get all red. All RR. For number 6, cross white with white, you get all white. WW. So for number 2, what if you cross red with pink? You get 2 red and 2 pink. For 3, red crossed with white. All 4 are pink. For 4, pink crossed with pink. You get is 1 red, 2 pink, and 1 white. And for number 5, pink crossed with white. We get 2 pink and 2 white. A dihybrid genetic cross involves 2 traits at the same time. In pea plants, yeah, we're back with pea plants. Tall is dominant to short, so we use a big T for tall and a little t for short. Let's throw in a second trait. How about color of the seed? Just for kicks, we'll say green seeds are dominant over yellow, so we use a big G for green seeds and a little g for yellow. With a dihybrid cross like this, there are 4 phenotypic outcomes. We could get tall plants with green seeds, tall plants with yellow seeds. We could get short plants with green seeds, or short plants with yellow seeds. So let's cross a heterozygous tall, heterozygous green plant, big T, big G, little g, with another heterozygous tall, heterozygous green plant, big T, big G, little g. So here's our genetic cross. First, we need to determine the different combinations of genes in the gametes for each parent. For the first parent, there are 4 different gene combinations. Big T, big G, big T, little g, little t, big G, little t, little g. For the other parent, likewise, there are 4 different gene combinations. Big T, big G, big T, little g, little t, big G, and little t, little g. These gene combinations represent gametes, reproductive cells. So now we're ready to draw our Punnett square. Since there are 4 different gene combinations or gametes for each parent, this Punnett square is going to be 4 boxes by 4 boxes, for a total of 16 boxes. We'll take the 4 gametes for the first parent and line them up along the side. 1, 2, 3, 4. We'll take the 4 gametes for the other parent and line them up across the top. 1, 2, 3, 4. Now we fill in the boxes where the rows and columns intersect. Be careful. This might take a few moments. Remember, I said don't get lost. Now you know why. So, in this box, big T, big G, big T, big G, big G, little g, big T, little t, big G, big G, big T, little t, big G, little g, big T, big G, little g, big T, little g, little g, big T, little t, big G, little g, big T, little t, little g, little g, big T, little t, big G, big G, big T, little t, big G, little g, little t, big G, big G, little t, big G, little g, big T, little t, big G, little g, big T, little t, little g, little g, little t, big G, little g. We're almost done. Little t, little g, little g. Whew, I think we got all the boxes filled in. They look right to you? Now we're ready to figure out the phenotypic outcome. For these dihybrid crosses, we're not too concerned about the genotypic outcome. It just gets a little crazy. So we're more concerned about the phenotypic outcome. In other words, we want to determine how many tall green ones, how many tall yellow ones, how many short green ones, and how many short yellow ones. So first, I'm going to look for tall green. I'm going to go through all 16 boxes, and if I see a tall green one, I'm going to circle it. Ready? Here we go. 1, 2, 3, 4, 5. Are they all tall green? Nope. 6. Nope. 7. 8. Nope. Nope. 9. Nope. 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 I count 9 tall green. You count 9 tall green? I count 9 tall green. Now we're looking for tall yellow. I'm going to go back through the boxes that I didn't circle, and I'm going to look for tall and yellow. I'm going to put a slash through the box if it's tall and yellow, so that I don't confuse it with the ones I just circled. Here we go. Tall yellow, tall yellow, tall yellow. I got 3 tall yellow. You got 3 tall yellow? I got 3 tall yellow. Now we're looking for short and green. I'm going to go through the remaining boxes, and I'm going to put a slash in the other direction if it's short and green. Here we go. Yep. 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 Nope. I got 3 short green. You got 3 short green? I got 3 short green. We only got 1 left, and it's short and yellow. So for my phenotypic outcome, I get 9 tall green, 3 tall yellow, 3 short green, and 1 short yellow. In a typical dihybrid cross, where both parents are heterozygous for both traits, our phenotypic outcome should show a 9:3:3:1 ratio. Let's try another one. In this case, we're going to cross homozygous tall, heterozygous green with heterozygous tall and yellow. So our cross looks like this. Big T, big G, little g, that's one parent. Cross with big T, little g, little g, that's the other parent. First, we look for different gene combinations in the gametes for each parent. The first parent has just 2, big G and big T, little g. We don't need the repeats. For the other parent, there are just 2, big T, little g and little t, little g. Again, we don't need the repeats. Never mind. So our Punnett square is going to be 2 by 2 for a total of 4 boxes. We'll take the 2 gametes for the first parent and line them up along the side, here and here. We'll take the gametes for the other parent and line them up on top, here and here. Now we fill in the boxes. Big T, big G, little g. Big T, little t, big G, little g. Big T, little g, little g. Big T, little g, little g. We're almost done. Just figure out the phenotypic outcome. In this case, we get 2 tall green and 2 tall yellow. Alright, ready for one more? This one's easy. How about big T, big G, crossed with little t, little g, little g? How many different gene combinations are there for the first parent? I see only 1. Big T, big G. How many different gene combinations are there for the second parent? Again, I just see 1. Little t, little g. Like I said, this one's pretty easy. Just 1 box. Take our big T, big G and put it on the side. Take our little t, little g, put it on top. Fill in the box. Big T, big G, little g. So our phenotypic outcome is 1 tall green. I know, we could sit here and do these all day. I might make you do that. But I'll let you go for now. I should put some extra just for fun problems up for you. Anyway, that's an introduction to genetics. I hope you had fun, and I'm going to leave it there for now. Till next time, remember, don't get lost. I'll see you then. Have a great day.